Good evening, and welcome to Brookline Booksmith tonight. My name is Jim Burley. I'm the events director. Thanks for coming. Thank you for your continuing support for your award-winning neighborhood independent bookstore. To keep up with the events in this series, check us out online at brooklinebooksmith.com. Pick up a copy of our schedule, sign up to be on our mailing list. You can do all this at the counters while you're buying Human Trials, which is on sale tonight, and which you all must purchase. We're very thrilled to have our neighbor Susan Quinn with us tonight. Her articles have appeared in Atlantic Monthly and the New York Times Magazine, among other places. She is a biographer extraordinaire, the author of two celebrated studies, Marie Curie, A Life, and A Mind of Her Own, The Life of Karen Horney. This is Susan's latest, Human Trials, Scientists, Investors, and Patients in Quest for a Cure, a remarkable look at the bumpy ride to find cures for rheumatoid arthritis and multiple sclerosis. With empathy and with edge, Susan maps the race with astounding inside access and with the clearest of eyes. The great novelist Chris Bohjalian wrote, "The same talents that make Susan Quinn such an immensely gifted biographer serve her well in this fine new book. She's a tireless researcher, a resourceful historian, and she writes individual scenes that have the pace and power one expects to find in a novel. Human Trials is absolutely fascinating, a window into the world behind the medical marvels we often take for granted, and their very human cost." Susan will read to us for a bit, take a few of your questions, and then be available to sign copies of this book. Please help me welcome Susan Quinn.
Thank you, Jim. It's a pleasure to be speaking tonight under the auspices of one of Boston's very best independent bookstores, the Brookline Booksmith. Because I know this is an audience of writers and of passionate readers, I'm going to talk first, before I read, about some of the writing issues I faced in this book. And then I'll read a little. I've been working on this book for the past 5 years. Actually, 5 years is a relatively short time for me. My biography of Karen Horney took 6 years, and the one of Marie Curie took 7 years. So I'm getting faster. And this book, Human Trials, presented very different challenges than the previous two books. Biographies have a natural shape, an obvious chronology, the story of a life. In fact, the challenge in biography, as Catherine Drinker Bowen once said, is to break the back of the chronology, to find connections by foreshadowing and by backshadowing between events, to avoid the tendency to tediously report what happened next and next and next and next in a life. In Human Trials, I was faced with an entirely different problem. From the start, I was clear that I didn't want to write the story of a lone scientist on the quest for a cure for disease X or Y. That would have been a lot like biography, but that wasn't what I had in mind. I wanted to tell the larger human story of all the people, and even the mice, who contribute to drug discovery. These people were in different places and didn't necessarily know each other, and their actions were often happening simultaneously or overlapping. The only thing that tied them together was the quest for a cure. And yet, I had to lay out the story in a way that would give the reader a sense of continuity and forward motion. The image that comes to mind is of a doll. Over here in the dollhouse is the laboratory with the postdocs and the mice. And over there is the clinic with the doctors and the despairing patients. Then along comes the venture capitalist. He's up here on some high floor of a Manhattan office building. And then you have the suits, the company people, the CEO and the vice presidents with their offices out on 128. And there are the doctors who are called investigators. And there are the patients in the trial who are struggling with illnesses in small towns and big cities all over the United States and Canada. It's a cast of thousands. And there are rooms in the house that are inhabited by concepts, which need to be explained as well. Concepts like autoimmune disease, and the experimental idea that underlies the quest in this book, an idea called oral tolerance. So there are all of these disparate things to explain and to connect in some way. How do you deal with this complexity? That was the challenge of this book. And how do you provide the reader with a guide rope? One way, a way that's often used, would be to introduce myself into the book as a character. And this is done many times successfully. A book I love is a book called The Spirit Catches You and You Fall Down by Anne Fadiman. And in this wonderful book, she is a character and a player, and interacts with the various people who are in her story. But I decided not to do that. I played with it, but ultimately decided against it. In other words, I don't tie the various stories together by saying, and then I went to New York and interviewed the venture capitalists. It would have added yet another layer to the story, and it would have put up an additional barrier between the reader and the story, which is the compelling story of the quest for the cure. So I've used instead a journalistic convention of simply putting the reader in the place, though all of you, as sophisticated readers, know that I'm there all the time shaping the story. That convention of staying invisible in the story was confining at times. For instance, there was a moment when I called one of the patients in a trial, and I had to tell her that the trial hadn't succeeded. And it was a very dramatic moment. She cried. And I would love to have been able to replicate, to just reproduce that exchange in the book. But since I wasn't a character in the book, I couldn't do that. So I had to use other devices, you know. So-and-so was, I don't know, drying her hair when she got the phone call which told her da-da-da-da. But the convention has many advantages. One, as I said, is the immediacy that it allows. And the other was that this story that I tell in this book took place over 15 years. And I was only there for the last 5. But using this convention, it's possible for all of us to pretend to be in this kind of conspiracy together, that we're there through the whole 15 years. And one reason that I was able to do this also, and to talk about the 15 years, is that I had the good fortune of having a main character, a protagonist, we'll call him, the principal scientist, who kept a journal and who was willing to share it with me. Now, this is a fairly factual journal. It's not particularly introspective most of the time, but it's cumulatively over 15 years, it's a wonderful document. You get a sense of what's involved in this process. So, but then, so I wasn't going to use myself, I decided. But I still had this question to answer: how to link the story so that the reader doesn't get lost in the complexity and is compelled forward. I didn't want to have any moments where the reader got discouraged and put the book down, saying, this is too complicated, this is too teachy, this is too didactic. And yet I had a lot of information to convey. Well, one of the things I did to solve this problem was to keep the book short. It's under 300 pages. And that was intentional, and I'm pleased about that. I also decided not to have endnotes, not to have bibliography, not to even have an index. A scholar friend of mine said, I never trust a book that doesn't have an index and a bibliography. But this book wasn't written for him. Another solution was to always clearly define for the reader which room they were in this imaginary house. And thus the chapter titles of the book. The first one is called, what is the first one called? I've forgotten already. The Clinic, the Disease, the Lab Director, the Researchers, the Big Idea, the Capitalists, so that each chapter is well defined, so the reader knows where he or she is. And then I kept as a touchstone and never strayed too far from the quest for a cure for the disease. Many people in this book were involved in some small part of that quest. For instance, I once went to a conference that the CEO participated in that Montgomery Investors was giving in New York, and he gave a presentation in which he never once mentioned any of the diseases that he was trying to find a cure for. Because it was all about other things. It was about the investment and business side of it. There's nothing wrong with that. It wasn't crass. It was just that was where he was at. But in order to keep the reader engaged, I wanted to stay with the fact that this was about diseases that affect and afflict human beings. Another thing was to have the characters have real characters in the book, but not too many characters, so the reader could come to care about them. And to have this one main character, my protagonist, Howard Weiner, whose story dominates and whom the reader can care about. And the book begins and ends with scenes of Howard Weiner in the clinic with patients. Now I'm going to read excerpts to you that take place in some of the various rooms in that dollhouse I was talking about. What I hope to convey by the end of the book is a sense of the way in which many actors, often at the same time, are participating in a vast enterprise for very high stakes. And I hope you will get a sense of the very human story and the tremendous heart that goes into taking a promising idea and turning it into a treatment for terrible diseases. In this case, the target of the effort is autoimmune disease in general, and multiple sclerosis and rheumatoid arthritis in particular. But my hope is that readers will come away from the book with a sense of how every promising idea for treatment travels from laboratory bench to bedside. Chapter 1, The Clinic. Most of neurologist Howard Weiner's days are spent in his laboratory or on the road, talking at conferences. But on Tuesdays, he takes the white coat, size 42 long, down from his office door, pulls it on over his blazer, and walks from his lab to the multiple sclerosis clinic at Brigham and Women's, a Harvard teaching hospital in Boston. On this particular Tuesday, it's windy and cold as he heads across the mall of Harvard Medical School, his white coat flapping at a walk that verges on a trot. Inside the hospital, he swings by the coffee shop and picks up juice and a blueberry muffin to be consumed between consultations, and hurries down the long corridor everyone calls the Pike. On the Pike, which connects the old and new Brigham Hospital buildings that have accumulated over a century, Weiner slows momentarily to meet and greet other white coats moving in the opposite direction. Then he hurries across the grand hospital lobby and through the neurology waiting area to the doctor's room, where he scans the roster of multiple sclerosis patients he will see that morning. As usual, he recognizes most of the names on the list. Many of them are long-term patients, and most of them are not doing very well. There are some very sick people coming in today, he says softly. Howard Weiner is a fit 6-footer with thick glasses that can make his eyes look wild. His hair used to be abundant and curly, but now that he's in his 50s, it's slicked down and thinning on top, adding prominence to his nose. He has a gleaming smile that flashes often, but he is incapable of irony or sarcasm. Because of his deep bass voice and his earnestness, one of his poker buddies thinks he would have made a good rabbi. Weiner, however, has chosen to devote himself to another mission, finding a cure for multiple sclerosis. Even after 30 years of searching, including many setbacks and frustrations, his zeal is undiminished. Weiner has always been an activist when it comes to treating multiple sclerosis. I think you've got to be doing something, he frequently declares. To buttress his argument, he sometimes quotes a line from King Lear, "Nothing will come of nothing." But the truth is that much of the time, multiple sclerosis defeats his interventions. That's why on Tuesday evenings, he so often writes in his journal about how tough his day in the clinic has been. He doesn't like to see people getting worse. The first patient on the roster this Tuesday morning is new to him. She is Harriet M., a 43-year-old from New Hampshire who is experiencing numbness in one arm and has come to get a second opinion. Harriet is a small woman with curly bleached hair that creates a halo around her face. She is wearing comfortable knit pants which accommodate her slender waist and full low hips, and a short T-top reveals a little of her firm midriff. Her eyes look puffy and tired, but her skin is smooth and youthful, and her arms look strong. She lifts weights, she tells Weiner, takes care of herself. Weiner listens to Harriet's story, a variation on hundreds of others he has heard in the MS clinic over the past 25 years. 8 years ago, she had a cold that resulted in dizziness and numbness on one side of her face. Her doctor referred her to an ear specialist, guessing that she probably had an inner ear disturbance related to the infection. But for 2 years after that, Whenever she looked at her feet, she got a strange sensation in her back. When her left arm began to get numb recently, she thought it might be because she had carpal tunnel syndrome from her job. That was when her doctor mentioned MS as a possibility. The story has revealed a lot to Weiner. Probably the facial numbness 8 years ago was a first MS attack. Most telling is the report that Harriet has been feeling shocks down her spine for the last 2 years when looking at her feet. This was probably the Lermite sign, caused by a lesion in the spinal cord that increases pressure when the neck is bent forward and down. It is a classic symptom of multiple sclerosis. So far, though, Weiner is going only on the patient's history. Now he asks her to sit on the end of the examining table so that he can run through the neurologic exam. Howard Weiner likes to come prepared. His family teases him that he never comes home to Colorado without a set of clothes for every kind of weather. He always carries an extra laser pointer with him when he's giving a talk, just in case. Similarly, Weiner keeps his white coat fully equipped for clinic days. On one breast pocket is the white plastic tag bearing the wavy crest of the Brigham, next to the words Howard Weiner, MD, Neurology. And inside there's a plastic eye chart and a tuning fork for testing sensations. A black rubber reflex hammer and stethoscope way down the lower right pocket, and there's a prescription pad lower left. His car keys, also essential, are dropped in at the last minute. Now he taps Harriet's knee with his reflex hammer. He runs his car keys along the arches of her feet, noticing that her right big toe turns up instead of down, as it would in a normal subject. He asks her to follow his finger with her eyes, looking for disconjugate eye movements, which indicate that the brainstem has been affected. Finally, Weiner watches Harriet walk, a slight bit unevenly, from one end of the corridor at the Brigham Clinic to the other. By the time he's finished the neurologic exam, he strongly suspects that Harriet has an MS lesion affecting the left side of her body, but he won't say anything until he's taken a look at the film she brought along. He carries the giant manila envelope down to the doctor's room and throws the stiff films up, 2 at a time, onto the view box. Very quickly, he finds a white spot on the vertical ropes of Bray that tells him she has a lesion on the left side of her spinal cord in the neck area and 2 other lesions in the brain. To Weiner, the conclusion is now unavoidable. This woman has multiple sclerosis. There are 3 drugs on the market that target MS. The ABC, Avonex, Betaseron, and Copaxone. The ABC drugs, as they are called in MS circles, have given new hope to patients, but they are not the cure. The drugs can reduce the MS attack rate by only about 30%, and they don't work in all cases. All 3 must be given by injection on a frequent basis, and there are sometimes side effects. In addition to the ABC drugs, chemotherapy agents are gaining acceptance for the treatment of certain forms of rapidly advancing MS, but these, although sometimes dramatically effective, have worse side effects than the ABC drugs. About 15 years ago, Howard Weiner got an idea for an entirely new and potentially revolutionary approach to multiple sclerosis. This approach, if successful, could result in a safe oral treatment not only for MS, but also for other diseases with similar autoimmune characteristics. From the moment he discovered that the approach worked in animals, he believed its development was going to be his crowning scientific achievement. The idea, called oral tolerance, was ridiculed in the beginning, but it has since become the focus of research in laboratories all over the world. So far, no one has been able to show conclusively that it works in treating human disease. Sooner or later, Weiner insists, it has to work. When that happens, I'll be a happy man. For the New Hampshire couple who have come to see him on this Tuesday, however, such dreams of future breakthroughs are no comfort. Weiner enters the room where Harriet and her husband are waiting and hoists himself onto a swivel stool. With his hands clasped around one knee, he turns to face the couple. When you have only one symptom, he begins in his low, ponderous voice, then it's difficult to know if it's MS. But when a number of symptoms point in the same direction, as in this case, it becomes possible to make a diagnosis. Then, incongruously, Weiner offers the New Hampshire couple a big city analogy. When you see the Empire State Building and Central Park and Madison Square Garden, he tells them, you know you're in New York. Neither of them smiles or even seems to hear what Weiner has just said. But then, as he starts to speak again, the woman stops him. Wait a minute, she says in a trembling voice. You say you think I have MS. It's not for sure? No, Weiner answers firmly, it is MS. I have no doubt. He continues, MS is an autoimmune disease of the nervous system in which some of your cells attack your own tissue. You probably had a first attack 8 years ago, and that's lucky, because there has been a long interval between attacks. But with MS, it's impossible to know the future. It's a little like being in a boxing ring where you keep getting hit by an invisible opponent. You may be in the ring with a 5-year-old who doesn't hit too hard, or you may be in there with a pro who does. None of this is registering with the patient and only a little with her husband. Do you understand? Weiner asks. Well, right now, her voice trails off. She is staring at the floor, about to cry. Understanding will be a long and complicated process. So that introduces you to my protagonist and to MS, one of the autoimmune diseases that this book is about. Now I want to introduce you to the lab and to the characters. One of the characters in the lab, a woman named Ruth Maron, Israeli woman whom I spent a good deal of time with. You will be able to tell, even though, again, I don't identify myself as there, that I was with her during these events I'm reading about. Ruth is used to tending the animal experiments. She does it on Saturdays and Sundays because it's a long trip into the labs for her technician. But even when the mice are producing great results, she is still troubled after all these years by the animal work. I find it stressful, she says. I don't like treating living beings as test tubes. With a sad smile, she tells of her son's announcement that he was going into computers instead of biology because, quote, you've already killed enough animals. Ruth and others use the word sacrifice to describe the killing of animals in their work. It is a euphemism that contains a justification. The animals are sacrificed for the good of humankind. What makes animal research acceptable to Ruth and the others involved in it are the results. Animal research has produced cures for many scourges, including diphtheria, tuberculosis, and polio. Almost everything that is now known about autoimmune disease has come from animal studies. There is a paradox, however, at the center of animal research. It is because the bodies, the brains, and sometimes even the psyches of animals are like ours, that they make good models for the study of human ailments. And it is because the animals are like us that we have qualms about using them in this way. 50 yards or so on the other side of Longwood Avenue, Ruth enters into another world, the animal room in the Warren Alpert building, a cool gray granite addition fused onto one wing of Harvard Medical School. Security is tight here, but Ruth nods to the guard, who knows her well, and swipes her plastic card to unlock the door to the lab. The protests of animal rights activists have made such precautions necessary. Also, partly because of the pressure from animal rights groups, the cages are large and well maintained. Each of the thousands of mice in this facility was purchased for around $15 and is maintained at about 33 cents a day. Ruth describes the facility, in fact, as a 5-star hotel. Yet, it is definitely a rodent hotel. From the moment Ruth passes through the secured door, the animal smell is thick in the air. In a dressing room, Ruth strips to her underwear and pulls on blue surgical scrubs over her slim figure, slips disposable booties over her shoes, and a disposable cap over her ponytail. All of this is part of the effort to keep pathogens away from the animals. In the control room, the attendant, who monitors entries and exits from behind glass, tells Ruth that there was a flood in one of her cages and the animals died. I don't think it was one of the critical experiments, she says, but we'll see. Using her card one more time, Ruth opens the door into a room of stacked cages filled with hundreds of mice. There are many kinds of mice here, some black, some white, some with large litters of tiny blind pups, others with older babies nursing at their mother's tits. Each cage has a clean lining of cedar chips, and each has a card taped to it that tells a story, what breed the mice are, who the researcher is, and what's being done to them. Ruth sets down her supplies, pulls on thin gloves, collects her thoughts, and reaches for a cage to begin her work. Swiftly, she grabs the pink tail of one of the little white mice. Then, with her other hand, she takes hold of it firmly by the neck. She wants to see if the mice in this group are showing signs of EAE, which is the mouse equivalent of MS. The tail is a good indicator of disease, since paralysis sets in there first. Some of the mouse tails curl actively around Ruth's finger, indicating that they are healthy. But other tails are listless, and some of the mice are dragging whole rear ends behind them, using their front feet. Ruth observes and assesses each mouse, using a scale in which 0 means no disease and 5 means death. She looks at their fur, which turns scruffy and loses its lustrous white with disease, and she turns them over on their backs to see how hard it is for the little creatures to right themselves. There are some who have become sicker and others who are getting better. In short, the mice have ups and downs similar to those experienced by human patients with relapsing or remitting multiple sclerosis. Before she leaves, Ruth sprays water on the food pellets of the afflicted mice because some of them have a hard time getting to their water bottle. In the animal room, Ruth works alone. But back at the lab, she almost always has company. Usually the 2 postdocs she is working closest with at present, Anthony Slavin from Australia and Gabriela Garcia, who comes from Brazil. She tells them the good news that the diabetic mice seem to be contracting EAE. This is good news. She tells them about the flooding, which fortunately didn't wreck any experiments. Conversation then turns, as often happens, to mice in general. Anthony's progress, it seems, has been slowed by a major cell death, which has forced him to start over breeding a new batch of mice. And Ruth remembers that when she was finishing her PhD, all the mice got some terrible disease and had to be sacrificed. She was devastated, but fortunately she had an easygoing advisor who told her, well, this way you finished your PhD sooner. The kind of genetically engineered mice these researchers use are hard to replace. Bred through sophisticated processes on experimental mouse farms, they aren't always available in desired numbers. The Center for Neurologic Diseases can rarely fill its standing order. Yet, without them, the work can't go forward. In the hierarchy of the lab, the mice may be at the bottom. But like most underlings, they are what keeps the whole operation going. The mice, Ruth Marone says, laughing, are more important than we are. Well, then we move on in my story to discussion of this big idea, oral tolerance, which turned out to be a very promising idea for treating autoimmune disease. The animal work showed that this approach might be a very promising one to try. And as soon as that happened, Howard Weiner started thinking about the next step. I'm going to jump way forward to the time at which it really seemed from limited human trials that this was an idea that needed to be tried out in major clinical trials. And at that stage in the process, it becomes necessary to find a lot of money. NIH grants can support the early human trials, but when it comes to the large clinical trials, you need millions. And the way you get those millions is to go to venture capital. And particularly since 1980, when it became possible for institutions and hospitals in the public sector to form partnerships with venture capitalists, this has been the way a lot of research, most research, has come to clinical trials. I'm going to move to the chapter that's called The Capitalists and just describe the 2 members of the CW group who did the initial funding of this and who organized the major fundraising that went on when they did a roadshow and raised $40 million. So these were the first capitalists that got involved. One of them was a man named Walter Channing, who had connections to the Brigham and Women's Hospital through his illustrious medical family, and who was a very adventurous investor. And the other was a man named Barry Weinberg, and the 2 of them formed the CW group. Walter Channing and Barry Weinberg, the C and W of the CW group, have been partners since 1968. They are an improbable duo. Channing, scion of a distinguished Boston family, grew up in Dover, a genteel suburb of Boston, attended Milton Academy and Harvard, followed by Harvard Business School. At the same time, Barry Weinberg was growing up in nearby Roxbury, a then Jewish neighborhood of modest triple-deckers. After Boston Latin and exam school, he graduated from MIT in electrical engineering. I had an uncle who said electronics. Weinberg explains. What did I know? It turned out he hated engineering. So before long, he decided to go to business school, choosing NYU over Harvard because he got a scholarship. Venture capitalists are gamblers who count on a lot of failures and a few big wins. Even though the industry has grown enormously since Channing and Weinberg began, its assets are a fraction of those available from commercial banks. Venture capitalists, however, are willing to take risks that banks won't, and to work as partners with entrepreneurs in getting their ideas launched. As venture capitalists, Channing and Weinberg turned out to have complementary skills. Channing had good instincts about what to invest in and was good at starting from scratch and putting a deal together, raising money from people he knew and who knew him. Weinberg was good at managing and believes that their hands-on approach during the startup phase of new companies has been the secret of CW's success. Weinberg is very methodical, according to Channing, who is not. Weinberg's office, just a few yards from his partners, could be in another building from Channing's. Channing's unwalled space is bathed in sunlight, and his windows frame a charming row of brownstones along 61st Street. Weinberg's walled office is dark and overcrowded. There are files piled everywhere. His windows overlook the harsh black glass and brass canopy of Trump Plaza, home of a large paint and wallpaper store on 3rd Avenue. Like Channing, he has his own art on the wall. A strange, dreamlike oil painting of a man in an empty room. A small, trim man in a well-made suit, Barry Weinberg stands with his back to the door, staring out on 3rd Avenue and talking on the phone with someone who wants him to invest in a startup company. Do you have data? Barry is asking the petitioner. As he listens to the long answer, he turns and riffles the pages of the proposal under discussion, which is sitting on his desk. Clearly, he is not impressed. We've been in the venture capital business for 30 years, Weinberg tells the caller, and we invest in the health field only, so we have a lot of experience. How is this different than, better than, previous technology? Another long answer. Your capitalization strategy, I think, is a little naive, says Weinberg, then adds, without much conviction, I don't mean to be demeaning. Weinberg tells the caller to send more data and agrees reluctantly to a lunch date in New York. We see about 1,500 investment opportunities a year, Weinberg says. We typically make 4 or 5 investments. When companies have failed, according to Weinberg, it has often been because of the inflexibility of the leading scientist or executive. We have become very, very sensitive to the nature of the person we back, he says. We've seen many projects over the years in which we were fascinated by the technology and the ideas, but did not feel the scientist was suitable for us. The course of drug development is never predictable, Weinberg explains. Unforeseen and untoward things happen along the way, and you've got to deal with them. And that means not being stiff-necked, not only knowing your stuff, but being able to sit down and talk with people and listen. Howard Weiner impressed Weinberg as the kind of person the CW Group could work with. An honest person, a good scientist. He didn't have a lot of respect for the commercial world, but he observed and was willing to adjust. And so began the partnership of the CW Group and Howard Weiner. Out of that partnership, a CEO was hired for the company, and it was a little like the farmer in the dell. You know, the CEO takes a vice president, and he takes another vice president, and before you know it, you've got 6 or 7 employees. And then Howard Weiner, the principal scientist and the CEO, went out on what's called the roadshow to places all over the world where there were investors willing to listen to their story, and sat in private rooms over very fancy meals and explained their promising idea for treating autoimmune disease, this idea of oral tolerance. And they raised on that first go-around, that first roadshow, $39 million. And then they had an initial public offering. Stock went up and down, as they do with these unknown ideas. It went as high as 15, I think, at one point, maybe even higher. But the truth was, as everybody knew, nothing would really be known until there were clinical trials and until there were results from clinical trials. And here's where my story really broadens and widens out to include many, many people. This trial, the first trial they had in multiple sclerosis, was a trial that involved 500 patients, and in sites, I think there were 14 or 15 trial sites all over the United States and Canada. And at each of these sites, there was a doctor, a principal investigator, who was recruiting patients to the trial until they had these 500 patients. Then they had a 2-year trial. Just give you a sense of what a long process this is. So then there's a 2-year trial, at the end of which there will be results. I'm not going to give away the end of the story, but I'm going to read you one last excerpt about a patient in the trial, actually the very last patient to enroll in the Mylarol trial. So here that is. Entirely by chance, the last patient to enroll in the Mylarol trial and the last patient to finish was a 29-year-old graduate student named Nicholas H., who lived in Montreal. So the last of several thousand of the MRI films that were taken in the Mylarol study didn't have to be packed up and sent by air. Instead, it was hand-carried down the hall. I should explain that the MRI films were all analyzed in Montreal. So this last patient happened to be in Montreal. Nicholas H. grew up in Greece and arrived in the northerly climate of Montreal at the age of 17. He sometimes wondered if he might have avoided MS altogether if he hadn't left home. A marathoner who had his first attack after a long run, he wondered too if the running might not precipitate attacks, or if the infectious mononucleosis he contracted shortly after arriving in Canada might have something to do with it. Most of all, he wondered about his future. Nicholas was a tall young man with a handsome aquiline profile, a radiant smile, and large brown eyes with long lashes that had a slightly stunned look in them. Even though he came from the Mediterranean, he welcomed the cold of Montreal winter and was well prepared for it, moving quickly along ice-layered sidewalks in his hooded jacket. "This is my ideal time," he explained. "All my attacks have been in the summer." The summer after he entered the Mylarol study, Nicholas didn't have an attack. That made him hopeful, and he took up running again. But then he had 2 attacks in fairly quick succession, and that caused numbness in his legs. After that, he hoped he was on the placebo, the sugar pill, and not the real drug. Like everyone else in the trial, he wanted to believe that Mylarol could change his life. Nicholas's graduate work was in experimental psychology, so he studied the brain and was able to follow developments in MS research. But that didn't make it any easier to know the future and what it would bring. Because of his MS, he felt more pressure to finish his PhD and get established somewhere. He wanted to remain in academia because it was what he loved, and because he believed the academic community would be tolerant of someone with a disability. Yet he sometimes thought it would be wiser to get a job in the pharmaceutical industry, because that way he could make more money quickly and save up in case he became disabled. "I mainly get sensory symptoms," Nicholas explained, "but what if the next symptom was a motor symptom and I couldn't work very well? You always think about that." On Tuesday, March 18, 1997, at a little before 2 in the afternoon, Nicholas H. walked up the hill to the MNI, the Montreal Neurological Institute, the limestone fortress Penfield built, and headed for the MRI unit. There he climbed up onto a narrow concave table and lay perfectly still as a technician slid a white plastic basket resembling a bird cage over his head. Then the table slid slowly into a tunnel so narrow that he could feel the sides of it along his arms and hips. There was a microphone inside the space so patients who became claustrophobic could ask for help. But Nicholas, the calm scientist, managed his session in the dimly lit MRI tunnel without too much difficulty. He lay immobilized in the tubular prison, waiting for the noises of the MRI, created by the magnet's pull on the machine's metal parts, to begin. First came a tock-tock, the sound of a ball-peen hammer tapping somewhere near his head. Then a louder, steadier sound, like a pneumatic drill doing double time. Then silence, the ball-peen hammer again, and other sounds, this time more like the alarms that plastic security tags set off at the door of a department store. Then doubling of that sound so that it began to resemble a Philip Glass piece. After 45 minutes, the set of films of Nicholas' brain was complete, and the Mylarol trial was officially over. At long last, the researchers conducting the trial could take off their blinders, and all those who had placed their hopes in Mylarol could find out if it worked. I'm going to end there. Thank you. Can we go right away? Um, the question. Well, Howard Weiner likes to say when confronted with the difficulty of finding a cure that the low-hanging fruit has been picked. In other words, autoimmune diseases are one of the very complicated challenges. And I don't think anyone can answer your question, really, how far the cure is away. I think I'll leave it there. I have to also remind you that I'm a journalist, so I was on a radio program where people were calling me up and asking me advice about what drugs they should be taking. And I had to make a disavowal. Yes, Ben in the back.
The story represents an interesting issue between the scientist as a pure research scientist and entering the business world, and the realm of the business world and the science world, the university and the business for profit, not for public use, etc. How do you explore that, and what have you learned about that issue?
I think in many ways the book is about how money affects science. Howard Weiner is someone like all research scientists who has had to struggle to get grants throughout his career. And this, as you and I'm sure others here know, is a very time-consuming and discouraging and aggravating process. And then there's that kind of money. There's one point in this story where Howard Weiner has been turned down for a grant and is feeling aggravated about it. And at the same time is talking with these venture capitalists. The grant was, I think, maybe for $30,000, can't remember. The venture capitalists are talking in the millions. They only talk in millions. So they're talking 3-4 million just startup to do a few little experiments. So, this is very seductive. Also, the money's necessary. These large clinical trials couldn't be done without it. At the same time, when you have venture capital involved in science, the clock is ticking. These are people who want a return on their investment. And no matter how much they claim they're willing to wait, there's pressure to move quickly. My feeling, after writing this story, is that probably the pressure of the clock ticking caused some decisions to be made that shouldn't have been made. I think they probably moved too quickly. I think everyone would agree at this point, in retrospect, that they moved too quickly into phase 3 trials. This remains a very promising idea, and there's ongoing trials now using this, and it may be that in fact it will turn out to be a successful treatment for MS. But I do think that the effect of the venture capital was to move things quickly into phase 3 trials. Also, I explore the whole area of the interface between the company, Autoimmune Inc., the small biotech company, and the physicians who are on the front line with patients. And the way there is a kind of pressure for physicians, first of all, to recruit patients. They get paid by the patient. I never was able to find out. I got inside a lot, but I never did actually get a hard figure on how much Autoimmune was paying per patient in this trial, partly because they pay different amounts to different places, depending on whether it's a prestigious research institute or whether it's a physician in private practice. So there's money involved in recruiting patients. There's money involved in keeping the patients in the trial. There may be times when their rescue medications might be appropriate, but the doctor might not want to use them because it would mean that the person would have to be taken out of the trial. Patients should know, by the way, that they always have the option of coming out of a trial, but that creates certain kinds of pressures and tensions. There's also the interesting tension between the company who want to keep the trial clean and don't want the doctors using all kinds of extra medications to help their patients, and the doctors who by and large think in terms of helping individual patients. So the vice president of Autoimmune one of them said to me at one point, you can't think about individual patients when you're in this business. You think about the greater good in the long run for the large number of people. But doctors should think about, as you know, individual patients. So that's a tension. There are a lot of tensions in these interfaces. There's another tension about information and whether it should be revealed or not. The tradition in academia is that you tell all, and in fact your whole reputation rests on having other people replicate your research. So you need to publish your work and to have it tested by other researchers. And that's the way that world works. And the coin of the realm in research is really reputation. The coin of the realm in business is money. And that means patents. And patent lawyers want to protect information. So there have been numerous instances in which researchers have been put under pressure not to reveal the results of their work because it would be bad for the company. And sometimes they have entered into agreements which actually silence them. And this is a bad thing, no doubt. I must say, I was appalled that a Wall Street Journal review of my book used it as an argument for no limitations on drug prices and for just total justification of big pharma's large prices, etc. And I don't think my book is an advocate of that. But what I did in my book, I didn't look for the bad guys. If I'd wanted to do an investigative book about people who were abusing the system and who were being swayed or influenced in very serious ways by money, I wouldn't have chosen Howard Weiner. I wouldn't have chosen these people. These were dedicated people. And there may have been some what they call dry labbing, which is sort of inventing patients or results. I mean, that might even have happened with these trials. It does happen with others. So that's a key question. Money runs through the book. It's all over the place.
My wife and I thoroughly enjoyed the book. I think you just did a fantastic job of capturing oral tolerance and Dr. Weiner. And I have one request and one hope. The request is that you continue to follow Dr. Weiner and the progress of oral tolerance. And my one hope is that you go back into the journal and you learn more about his poker playing so that in the future I can beat him.
This is one of his poker buddies. Not the one who said he should be a rabbi, though. I'd like to acknowledge that Dr. Howard Weiner is here tonight, and I'm very pleased that he is. Howard, he's standing over there. I'm not sure that's going to be on television or not. But the next to last chapter of my book is called The End, and the last chapter is called The Beginning. And that is because even though there were some disappointing results in this particular trial, there are very many other promising developments since then. And I'm hoping in the paperback edition of the book that I'm going to be able to have an addendum that says, we got oral tolerance to work in human disease. I think there's no doubt it's going to happen. And there are trials ongoing. Teva, an Israeli-based company, is now doing a very large trial in multiple sclerosis. There's some very promising findings also in Alzheimer's disease, and there are some positive findings in diabetes now. So all told, there is reason to keep watch. Stay tuned.
I'm curious as to how you latched on to this particular idea, where it came from. Was your original intent to sort of profile human trials, or did it really initiate as more of a biography?
I guess the person came first, and then the idea of the book came after. I had known Howard Weiner he was a friend of a friend, and I heard him talking one night about, he was just in the process when I happened to have dinner at someone's house, and he was talking about how he was raising millions of dollars for this very, very promising idea. And it was actually my husband who said, "That's your next book." I didn't think so, but he always winds up being right. And what made it very appealing was that Howard was willing to become very open with me once he decided to do it. It took him a long time to decide to do it. But once he decided to do it, we really had a good agreement, a signed agreement that I could write whatever I wanted to write. He had this, what I think is a very admirable scientific attitude, which is the facts are there and you can do with them what you will. The only proviso was that he could correct facts that were wrong. So I had freedom in that way, and I also had access to these journals, and that was quite remarkable to have that access. And then also, because he had agreed to cooperate, he opened doors for me at the company. So for instance, on the morning when they got an announcement of negative results, I was supposed to be there that morning at 9, and one of the vice presidents said to Howard, maybe we should tell her not to come, and he said no, she should come. So I was there when they announced these results, I was there when they told the employees that there were going to be layoffs. I was there for all that painful stuff, and that, I think, is what makes the book powerful, I hope.
Okay, thank you very much. Big hand for Susan.
Thank you. Thank you.
Pick up a copy of Susan's book, get it signed. Thank you, everyone, for coming tonight. Thank you, Susan.
Thank you.
Only I can read. Good evening, and welcome to Brookline Booksmith tonight. My name is Jim Burley. I'm the events director. Thanks for coming. Thank you for your continuing support for your award-winning neighborhood independent bookstore. To keep up with the events in this series, check us out online at brooklinebooksmith.com. Pick up a copy of our schedule, sign up to be on our mailing list. You can do all this at the counters while you're buying Human Trials, which is on sale tonight, and which you all must purchase. We're very thrilled to have our neighbor Susan Quinn with us tonight. Her articles have appeared in Atlantic Monthly and the New York Times Magazine, among other places. She is a biographer extraordinaire, the author of 2 celebrated studies, Marie Curie, A Life, and A Mind of Her Own, The Life of Karen Horney. This is Susan's latest, Human Trials, Scientists, Investors, and Patients in Quest for a Cure, a remarkable look at the bumpy ride to find cures for rheumatoid arthritis and multiple sclerosis. With empathy and with edge, Susan maps the race with astounding inside access and with the clearest of eyes. The great novelist Chris Bohjalian wrote, "The same talents that make Susan Quinn such an immensely gifted biographer serve her well in this fine new book. She's a tireless researcher, a resourceful historian, and she writes individual scenes that have the pace and power one expects to find in a novel. Human Trials is absolutely fascinating, a window into the world behind the medical marvels we often take for granted, and their very human cost." Susan will read to us for a bit, take a few of your questions, and then be available to sign copies of this book. Please help me welcome Susan Quinn.
Or why? That would have been a lot like biography, but that wasn't what I had in mind. I wanted to tell the larger human story of all the people, and even the mice, who contribute to drug discovery. These people were in different places and didn't necessarily know each other, and their actions were often happening simultaneously or overlapping. The only thing that tied them together was the quest for a cure. And yet, I had to lay out the story in a way that would give the reader a sense of continuity and forward motion. The image that comes to mind is of a doll. Over here in the dollhouse is the laboratory with the postdocs and the mice. And over there is the clinic with the doctors and the despairing patients. Then along comes the venture capitalist. He's up here on some high floor of a Manhattan office building. And then you have the suits, the company people, the CEO and the vice presidents with their offices out on 128. And there are the doctors who are called investigators. And there are the patients in the trial who are struggling with illnesses in small towns and big cities all over the United States and Canada. It's a cast of thousands. And there are rooms in the house that are inhabited by concepts which need to be explained as well. Concepts like autoimmune disease and the experimental idea that underlies the quest in this book, an idea called oral tolerance. So there are all of these disparate things to explain and to connect in some way. How do you deal with this complexity? That was the challenge of this book. And how do you provide the reader with a guide rope? One way, a way that's often used, would be to introduce myself into the book as a character. And this is done many times successfully. A book I love is a book called The Spirit Catches You When You Fall Down by Anne Fadiman. And in this wonderful book, she is a character and a player. And interacts with the various people who are in her story. But I decided not to do that. I played with it, but ultimately decided against it. In other words, I don't tie the various stories together by saying, and then I went to New York and interviewed the venture capitalists. It would have added yet another layer to the story, and it would have put up an additional barrier between the reader and the story, which is the compelling story of the quest for the cure. So I've used instead a journalistic convention of simply putting the reader in the place, though all of you, as sophisticated readers, know that I'm there all the time shaping the story. That convention of staying invisible in the story was confining at times. For instance, there was a moment when I called one of the patients in a trial and I had to tell her that the trial hadn't succeeded. And it was a very dramatic... Thank you, Jim. It's a pleasure to be speaking tonight under the auspices of one of Boston's very best independent bookstores, the Brookline Booksmith. Because I know this is an audience of writers and of passionate readers, I'm going to talk first, before I read, about some of the writing issues I faced in this book. And then I'll read a little. I've been working on this book for the past 5 years. Actually, 5 years is a relatively short time for me. My biography of Karen Horney took 6 years, and the one of Marie Curie took 7 years. So I'm getting faster. And this book, Human Trials, presented very different challenges than the previous 2 books. Biographies have a natural shape, an obvious chronology, the story of a life. In fact, the challenge in biography, as Catherine Drinker Bowen once said, is to break the back of the chronology, to find connections by foreshadowing and by backshadowing between events, to avoid the tendency to tediously report what happened next and next and next and next in a life. In Human Trials, I was faced with an entirely different problem. From the start, I was clear that I didn't want to write the story of a lone scientist on the quest for a cure for disease X. At the moment, she cried. And I would love to have been able to replicate, to just reproduce that exchange in the book. But since I wasn't a character in the book, I couldn't do that. So I had to use other devices, you know. So-and-so was, I don't know, drying her hair when she got the phone call which told her da-da-da-da. But the convention has many advantages. One, as I said, is the immediacy that it allows. And the other was that this story that I tell in this book took place over 15 years. And I was only there for the last 5. But using this convention, it's possible for all of us to pretend to be in this kind of conspiracy together, that we're there through the whole 15 years. One reason that I was able to do this also, to talk about the 15 years, is that I had the good fortune of having a main character, a protagonist, we'll call him, the principal scientist, who kept a journal and who was willing to share it with me. Now, this is a fairly factual journal. It's not particularly introspective most of the time, but it's cumulatively over 15 years. It's a wonderful document. You get a sense of what's involved in this process. So, but then